Oh yeah, the various types of validity, make sure you review those. Construct validity, internal validity, external validity, ecological validity. And so statistical conclusion validity meant you're using the correct statistics for the type of data that you collected. Ecological means that the way that you had your experiment set up accurately reflects how that thing that you're studying occurs in the real world. So if you have a very realistic experiment, it has ecological validity. That's a specific type of external validity. Remember, external validity is all about generalizability, being able to say that what happened in your experiment is true outside of your experiment as well, and out in the real world. And if you did this again, you would find the same thing. Internal validity just means that within your experiment, if you found an effect that was a real effect, so you did actually manipulate what you meant to manipulate and measure what you meant to measure. And then let's see, construct validity, that was, let's see, what was that one? Refresh my memory. What was construct validity? That's right. Yeah. So your operational definitions are actually good operational definitions. That's construct validity. Sorry, it's been a long day already. Okay. And then threats to internal validity. So these are things that can produce an effect aside from your manipulation. So these are different types of things that would actually be confounds, like something that affects your participants outside of your manipulation. And we talked about a bunch of different types of those. Several times throughout the semester we've talked about this regression to the mean phenomenon, so that's going to be important to know. So what was regression to the mean? Yeah, if you get extreme scores with one measurement, the next measurement is likely to produce scores that are less extreme. Right? So one example that we used of that was in a more recent chapter. We were looking at reading ability in these students, and we said that if you select people that get extremely low scores, then when you test them again later, it might look like they improved a lot, but that's just regression to the mean, because you happen to select the people that just got really low scores that first time around. And that might have been a fluke that first time. It wasn't an accurate representation of how they perform on average. And then you all should know what attrition is. That's just people dropping out of your study. Any questions about those threats before we move on? So onward to Chapter 6, Control Problems in Experimental Research. So the basic nature of between and within subjects' designs. So how many times do you measure or take measurements from participants in a between subject design? Yeah, I see you saying one time. Everybody is tested once, and you have different groups. So you'll have one group of people who are in your experimental condition and another group who's in your control condition. Right? And each person is only exposed to one level of that independent variable. And then how do within subjects designs work? So your participants are exposed to different levels of your independent variable. So you might have them go through a control period phase, and then they are exposed to another level. They have an experimental phase in that same participant, as opposed to they only do control or only do experimental. So, let's see. And y'all remember the Barbara Helms study? That was the one with the juror who was made up to be either really attractive or to look kind of average or dumpy. And so that one, it's important to know that that exemplifies a type of study for which you couldn't use a within-subject design. You would have to use between subjects, because if the people did this jury decision and they read all the details of the case, and then they did it the exact same case again with somebody who just looked a little different, they would get suspicious. They'd say, well, this is the same case I just rated. And you just couldn't do a within-subject design for that. So you have to use between subjects. The participants have to be naive when they complete the experiment. Let's see, methods used to create equivalent groups. You all should be familiar with these from when we've talked about them in class. Random assignment, pretty straightforward. You randomly determine whether they're in the experimental or control groups. Matching is when you measure something in advance and then assign them to condition based on that pre-measurement. So one of the examples that we used of that was that study in which they looked at the effects of sleep deprivation on interrogation. And so some people didn't get much sleep on average. They might only get 6 hours a night. Other people get 9 hours a night. And so sleep deprivation is going to affect those different people in different ways. And so they use that measurement, how much sleep they usually get, to assign them to different levels of the independent variable. Did you have a question?
Yeah, so we don't need to know about like counterbalancing and reverse counterbalancing, since you don't have that on it.
If it's not on the study guide, then it will not be on the test. There might be something, but, well, let's see. No. Yeah, if it's not on there, it won't be on the test, so yeah. I guess there's no counterbalancing on the cumulative final, so you don't have to worry about that. Okay. What were demand characteristics? That's an important one. Anybody remember? Yeah, that's exactly right. There's something about your experiment that tips your participants off as to what the hypothesis is. And then they might alter their behavior to try to confirm your hypothesis or try to go against your hypothesis, depending on if they are trying to be helpful or not. And so typically it's a good idea to hide the true hypothesis from your participants until the experiment's over, or you'll risk running into these demand characteristics. Right? Now that doesn't necessarily mean full-on deception, it just means that you're withholding some information until the end. And let's see, oh yeah, this one's split across a couple of pages there. Different types of subject or participant bias. We've talked about the Hawthorne effect. People behave differently because they know they're in an experiment. Any questions about those different types of bias? Okay. Moving right along. We've got about 13 minutes left here, and I think just a couple of... Okay, yeah. So we'll see if we can't get through this stuff. I know it's a lot of material, but we've got another review session on Monday, so if we don't cover something in enough detail today, we've only got 3 chapters to go over on Monday as opposed to 9, so we should have more time. Alright, so questions from Chapter 7, Single Factor Designs. This is another one where I'll give descriptions of experiments and you need to determine exactly what was the design that was used.
Really?
What's that?
Can you rate your examples on a scale of 1 to 10? Last time they were hard, like, I think I remember this.
Yeah, so let's see. What were the examples, the experiments I used last time? Oh yeah, I think there was one where I had talked about the study of the effects of caffeine on reaction time, and I mentioned that people might have been exposed to different levels of caffeine, and then if that were the case, you would need to determine, is this a single factor design or a multi-level design? And remember, if it just, you can have a single factor design that has a single factor, caffeine, at 2 different levels, right? So that's a single factor. And then a multi-level design might have one factor, but with more than 2 levels. So you don't just have high and low. You've got high, medium, or low, or anything in between or beyond that. So if you're looking at, say, 2 milligrams of caffeine, 5, 10, 15, 20, that's multiple levels, but just one factor that's being studied, caffeine.
I don't know if you can say, but was that the example where we're talking about like old people and their memory and like if they drink caffeine early in the...
That was a different one that we talked about, and I think we used that one to talk about placebo controls and double-blind studies. So they gave some people a decaffeinated coffee as the placebo control. Yeah, so that's similar type of subject matter, but different example. Yeah. So, and anyway, you'll just need to know what these different designs are. And remember, everything in this unit was all single-factor designs. A simple single-factor versus multi-level. And then in the next chapter, that's when we started to get into the factorial designs, where there are 2 or more independent variables. Right? But with these types of descriptions, those are that's going to be relevant to the single-factor designs. And remember, non-equivalent groups uses a subject variable as opposed to a manipulated independent variable. Independent groups uses a manipulated independent variable. Usually people are randomly assigned to the level of that variable. Matched groups just uses the matching procedure that I just gave the example of. And then repeated measures is going to be the within subjects designs, where people are exposed to different levels of that independent variable. Oh yeah, you also need to be able to identify the number of levels of the independent variable, which is going to be most relevant for these multi-level designs. So if it's a multi-level design, how many levels are there?
So is that, like you were saying, like the coffee levels would be low, medium, or high? So it would be 3?
Yeah. So that if it was low, medium, or high, that would be 3 levels in a multi-level design. So any more questions about those?